when we think about NAMs, often there's a question of, are we using NAMs to conduct safety studies to collect safety data, or is it for drug efficacy data? I think, it really depends on what you're trying to accomplish. But I'm wondering, anyone provide some examples focusing strictly on the safety aspect of NAMs toxicology? And then the next question will be more focused on drug efficacy and how can we be sure that we can trust an organ on a chip to determine how a human may, respond to a drug. So
Yeah,
first on
I can
safety.
start this one. Yeah. Um, I think predominantly the response has been you run something in an animal first, and that informs you on toxicology strategy, and then you go back and look at your organs of interest. And I do think this is a common and a needed approach because like the question in the chat box right now in the Q&A chat box talking about the biggest criticism they hear is from people being able to not assess systemic problems on a specific context of use type of chip or omics system or in vitro technology. So I think, you really do need the whole model to inform on toxicology and then backtrack from there. Until you have a linked, lab on a chip or human on a chip, we're not really going to be able to assess systemic toxicology, and we're not going to be able to predict what's going to happen if there's something like off-target toxicities that you wouldn't normally see when you're looking on a single organ level. So that's why I think the context of use in these NAMs is so important to make sure that the model that you're building has the appropriate cell types, the appropriate serums, the appropriate factors needed to show maybe immune-mediated response or generate a cytokine storm, which are very often hard to predict in animal models. So until we get something, a little bit more complex, but we build it stepwise and validate it each step of the way, I think that is going to be a huge knowledge gap. But, from my experience, it's kind of starting in a rodent model, letting that inform your decision, and then working backwards from there to either figure out mechanism of action or, de-risk in terms of what you're seeing in terms of toxicology.
On that
Yeah,
point,
Ray,
though, yeah, go ahead,
I
Zaher.
want to offer a I want to offer to a different perspective, maybe slightly than Dr. Atkins. See, one of the problems is that once a drug, an investigation drug, is deemed toxic or a little bit toxic in animals, it's completely abandoned.
Yeah.
It becomes radioactive. It's untouchable anymore. However, that drug can be perfectly safe in humans, and we have so many examples of that. So the issue is that's not talked about a lot is not whether we can develop effective medicine, but how many tens or hundreds or thousands of perfectly safe drugs in humans that we already prematurely abandoned just because of that litmus test in animals. I mean, by our current regulatory standards, Tylenol would not have been developed, and aspirin would not have been passed through. These are all drugs that are, hundreds years old or more. If you apply the regulatory standards right now for the requirement for animal testing or the one that's fading away now, you will not have these essential drugs that you and I have in our medicine cabinet today. So there's an insanity about how we have gone about this, and I don't know we have time today to delve into the history of this, but our policies and regulatory policies in particular should not be reactionary. In 1938, there was a reaction to a big disaster that killed 100 people at the time, that required animals now in the FDA modernization in the Food and Drug and Cosmetic Act of 1938 to test on animals, although these animals were not validated to begin with as a surrogate for humans. Right now, forward to 1962, where again we had an issue with a drug that was given as morning sickness drug to pregnant women that caused birth defect. Then we tagged on efficacy on animals as a requirement, again without serious analysis of the best model system. These type of reactionary policies yes, make us feel good about ourselves. They quench the public outcry. But at the end of the day, are these the right systems that we should be developing or relying on obsessively? And the answer is no. In 1979, there was a bill in Congress to develop centers for alternative. As one of my panelists said, this is not a new thing, right? 1979, that bill was crushed at the time by the NIH, to be very honest. So had we and our leaders embraced these type of investments, 50 years ago, we would have been in a different situation right now, and the NAM field would have been in a different place today. So very important to think about what we are missing, not only what we are achieving, and also every single drug comes with 16 pages of side effects, right? Could have we reduce that list of side effects that come with every therapeutic that's based on modeling in animals? I argue that the answer is yes.